Hi, just checking you. There's a good, you can hear me okay at the other side of the pond. Is the connection okay? Good, thank you for the thumbs up. It's a pleasure to do this pas de deux together with Ernest Turro. We both, like you, locked away in our rooms here in Cambridge. And we really want to tell you about the exciting journey we have made over the past 6 years. Just to take you into Cambridge, you see here the place where I'm locked away, what is going back to 1540, when Trinity College was established. And if you wonder where I sit, I just sit behind that blue spot on the first floor of the lodge at the college. So it's a pleasure to reach out to you from Cambridge and to take you into the work we have been doing over the past years. I think it's maybe good to put it in a historical perspective where the Sanger Institute, and I didn't join that initiative at that time, but in 1990, together with the Broad and others, started sequencing the human genome. Then we all know that the first variation was mapped to the genome through the initiatives of HapMap1 to HapMap3. And I really came on board in 2005 when we started preparing the first genome-wide association studies for the common diseases. So I really want to drag you to the green and the yellow bit, and particularly focus on the yellow bit. So the first part of my talk really will be on the pilot study for rare diseases, which is in a magnifying glass, was delivered through the NIHR bioresource and brought together data on 13,000 patients with rare diseases and their relatives. And it was really this partnership working with Ernest that made it possible to drive this big program across the country and also into the States and into mainland Europe. I want to round up with coming back how now the story of the rare variants with large effect sizes starts interplaying with common variants. And just want to show you one exciting summing up slide, what I think is going to play out in the next 10 years. So to start rare diseases, I think we all tend to forget that there are many, many rare diseases. And the most recent cataloging and shows that there are an estimated 10,000 rare disorders, and this number will continue to grow. In the UK, what has a 60 million population, we have an estimate that 3.5 million people experience ill health because of a rare disease in the first decades of life. And as you will be well aware, the estimates are not totally accurate, but we think that for half of those 10,000 rare diseases, the underlying genes have been resolved. So why the 100,000 Genomes Project? You may know that in the UK we have a rationed healthcare system, but it is free at the point of access. Which means that we have a good understanding of the current level of, you could say, failure to serve patients who have rare diseases. In this roadmap, it shows that it can take up to 5 years for parents before there is a diagnosis. With the old approaches, diagnoses were only reached in 1 in 10 to 1 in 5 cases, but generally closer to 1 in 10. And it created a lot of challenges for the family, as you can see, with isolation, financial distress, etc., etc. So the 100,000 Genomes Project has a number of very clear objectives. One is to reduce the diagnostic delay. We decided to not do this by a piecemeal approach, but we directly switched to whole genome sequencing because we recognize that eventually whole genome sequencing will become standard. And what is new, as you see in the green loop of the figure of 8 at the right, that as researchers, we were put in a position that we had to report back findings of variants that were relevant to clinical management. So it was no free ride of doing your research only. We had to establish these MDT teams, multidisciplinary teams in many different areas, and I will come back to that. What's unique, going back to the figure of 8, is the orange loop, is all the data that have been aggregated on the clinical phenotype, on the genotype, and data that are being collected through the National Health Service systems are available for research for anyone who wants to join. That means including, obviously, we would love to expand our collaboration with our colleagues in the States. So in this slide, just we wanted to share with you the sheer scale of the undertaking. We set up the pilot study, which did the first 13,000 genomes of the 100,000 that have been done by now. Engages 83 hospitals that are mapped out here. We have trained up to 360 clinical care teams, and we have engaged, to repeat, over 13,000 patients and their close relatives in the pilot study. The whole genome sequencing was done in a single establishment, which was managed by Illumina, and the clinical feedback was an obligatory task for the research community, supported by experts in clinical genomics. And then we obviously had the opportunity to do research. So let me take you into this pilot study. In this cartoon, you see how we work bottom up. We asked our clinical colleagues across the country which rare disease domains did they feel were deserving of more attention because they historically might have been ignored. So you see here the different rare disease domains to which sequencing capacity was allocated. And this was not a free ride. You got your sequencing done at no charge, but the clinical and laboratory phenotype data had to be deposited in a shared open database that is shared across the research community and is not protected by certain principal investigators. And I think Ernest and I are very proud of that we have changed the culture of compartmentalization of knowledge that was so normal in the rare disease research world. Being a hematologist, I would like to focus on those rare disease areas that come closest to me, and also if there are questions from our clinical colleagues or our quantitative colleagues, that is the area where I intellectually can most connect with. Particularly the bleeding, thrombotic and platelet disorders, which I've co-led with Ernest and others in the country, the stem cell and myeloid disorders, and for those who are in immunology, at the end, immune disorders are a disorder of the blood stem cell playing out through the immune system. So we would put that firmly under the umbrella of the hematopoietic system. And then at the end, there's an experiment on which I won't dwell, but Ernest might come back, is tapping in to this resource called the UK Biobank, which is 500,000 volunteers who have been genotyped and on who we hold, and you can hold, or you may have already sought access, have free access to all the data that have been generated. My daughter, and it is anxious times, and I'm sure for some of you listening that you're worried about the health of your family, but my daughter is on the front line of healthcare in Scotland and is looking after patients with COVID-19. But before that started, she asked me, Daddy, why do you do all this work on rare diseases? And I showed her this slide where in red you see the rare diseases of immune disorders, and in green with the bleeding, platelet, and thrombotic disorders. And I think we all realized that we had to completely rework the way we capture the clinical and laboratory information. Here we have worked with Peter Robinson, who was in Berlin, is now with the JAX laboratory in the States, to build on his work on the human phenotype ontology terms library, where you take as a doctor or a nurse the patient's story and code as the story comes to you without influence of clinical diagnosis. So you don't say, I think it is likely this patient has Glanzmann's thrombasthenia or Wiskott-Aldrich syndrome. No, you describe in computerized terms which symptoms and signs you observe in your patient. This is really, and Ernest may come back on that, helps you in gene discovery because it allows you to boost power if you have very data on large cohorts of index cases of rare diseases. So did this work? And I think I have to be honest, it is mixed. We feel it could have been better, but at the end, if you look here first at the heat map at the left, I highlight the 3 domains in red, stem cell and myeloid disorders, or SMD, in blue, the primary immune disorders, or PID, and in green, the area where we have particularly focused on bleeding, platelet, and thrombotic disorders, and on which I will come back. And you see, to no surprise, that if you have a bleeding, platelet, and thrombotic disorder, that the genes that are mutated in those patients at the somatic level do play out in other organ systems than only blood. And I think we all knew this, but it is now much clearer how vitally the gene that is not functioning appropriately plays out through the different organ systems of the body. This graph is very rich in information. At the top, you see the number of index cases enrolled per domain in the gray bars. At the right, you see the proportion of patients that have been given a particular term from a particular organ system. And at the right, you see that the PID domain that was driven by Hannah Lango-Allen, together with her colleagues, she clearly disciplined the clinicians to hand in a lot of data. And you see it's very comparable what Ernest has achieved. But there are other domains where we clearly have less rich data. And that is the reality if you start collecting data across 100 hospitals in the world. Some people would say here, and I just want to give you a warning that if you have mutations that give premature blindness, this is a cohort study of 800 cases from Moorfields General Hospital in London, that you only see that HPO terms are switched on for eye pathology. But this is not the case. This has to do with the high throughput of that ophthalmologists see their patients, and they tend to be very focused. They code what they see in the eye. They forget about the rest of the organ systems. So I think we need to realize that you can get artifactual observations if you have different clinical practices at the enrollment hospitals. Sorry, this slide is twice because this is the correct one, so I should have removed the one before. Sequencing, don't go to dwell on. I think when I was introduced, similar initiatives happening in America, obviously, often based on whole exome sequencing. We made a very clear decision that eventually it's going to be whole genome sequencing, and all the 110,000 patients of the 100,000 Genomes Project have been whole genome sequenced. I will, however, as a complementary technique that we developed earlier on, we made gene panel tests to sequence larger numbers of patients, and we did that at a time when whole genome sequencing was still relatively expensive. So I also show you something of the model of the right, where you have a gene panel test for 100 genes for a particular domain, and where you read around 1 million to 2 million bases. So a whole genome, and this is really for the nursing and clinicians that sit in the room, not so directly for the quantitative people, but just let's take you into your genome, roughly 20,000 genes, 3.2 billion bases, and it is this immense volume of data for which we had to develop complete new compute solutions that has been very much led by one of our colleagues in the bioinformatics team, who is not today joining the presentation. So let's look at data volumes. Here in the family where the mother carries the purple mutation and the father carries the green pathogenic variant, Liz has the autosomal recessive disorder, John carries the allele from the father that is disease causing, but because he only carries one allele, he has no disease, and I will come back to this later, and Sam got away with it. As I mentioned, all these DNA samples will be sequenced at a single sequencing center here in Cambridge, which is run for the National Health Service by Illumina, and 25 mobile phones would be streamed overnight to our supercomputer, and Ernest will come back on that, where then the variant calling and annotation was done of the genome of those 5 individuals. In the frieze that was underlying the manuscript that at the moment is present at bioRxiv, there were 176 million unique variants. So if a new genome would be sequenced from an individual we have not seen before, roughly 1,000 new variants would be observed that had not been observed yet in GNOMED, TopMed, or in the 100,000 Genomes Project. They come in 2 flavors. Focus here on the single nucleotide variants at the left, the insertions, the deletions at the right. May I draw your attention to the different Y scaling of the Y-axis, fitting to the rule that for every 10 single nucleotide variants, roughly 1 indel is observed. Most of the new variants that have been observed in one individual are belonging to the novel category. That means in your large databases in the States, those variants had not been observed yet. So how did we deal with these variants to make it tractable for the multidisciplinary teams to report back to the clinician who had referred that particular patient? And that is obviously by focusing on variants that were in the rare frequency space that were being reviewed by these MDTs. And that meant depending on the length of the gene list through which you filter the variants, the number of variants that had to be reviewed by the MDT was a handful. But if you look at those who work on neurodevelopmental disorder, it could be up to 20 variants in 20 different genes on which an opinion was required. We adhered to the American guidelines. We used a Congenica application developed by our colleagues at the Sanger Institute to deal with this virtual MDTs, bringing experts from across the world together to sit in a single room to review the results of the whole genome sequencing and to report back to the clinician whether a clearly pathogenic variant had been observed, which is causal of the disease, a likely pathogenic. And we deviate from the American practice that we do not report back the green variants because we feel that that causes a lot of confusion at the bedside when the clinician discusses the results with his or her patient. Ethnicity mapping was ethnicity allocation of the person who had been sequenced was obviously essential to take care that the correct control data sets were being used to look at the allele frequency of the variant that was going to be reported. And also family structure was computed automatically to first cousin removed. And I just want to draw your attention that we gave freedom to the principal investigator to decide on the best design of the study. And you see here, for intrahepatic cholestasis of pregnancy, only the mothers were, the genome of the mothers was sequenced. While if you take stem cell and myeloid disorders, which concerned children generally who couldn't make any red blood cells, frequently father, mother, and child were sequenced to speed up the process of reporting by a trio analysis approach. Gene lists. We have curated 10% of the genome together with our colleagues at the European Bioinformatics Institute, what is the sister institute of the NCBI here in Cambridge, but based in Cambridge. What you see in this map is that if we go from bottom left, where we have neurodevelopmental disorders, a gene list of 1,400 different genes. We go to the extreme at the right bottom, this rare disorder of pregnancy, ICP, where we had a single gene on which we reported. So what are the outcomes in terms of yield? Let's look at the first one, the HCM, what is hypertrophic cardiomyopathy, coordinated by our colleagues in Oxford. All patients were pre-screened on the diagnostic panel for the National Health Service. And if they were positive for a variant in a known gene, they were not enrolled. They did a good job. They didn't have a single variant in one of the known genes that was causal. Let's flip to the other side. How good can it get? And I mentioned to you this collection from the specialized hospital Moorfields in London with people who had premature blindness. Before we started the project, 2 in 10 patients would get a conclusive diagnosis. We are now crawling to 6 out of 10 patients that get a conclusive diagnosis. And we have not yet explored the non-coding space in this disorder, and Ernest might come back on that in his talk. Let's then look at neurodevelopmental disorder, 1,400 genes, a large and wide spectrum of pathologies. 1 in 3 patients get a conclusive diagnosis. But I would like to draw your attention to the paper of Meyer et al. In Nature Genetics to show how reaching a molecular diagnosis can change the life of children, where children were institutionalized, were cured by deep brain stimulation, highlighting that reaching a molecular diagnosis truly can transform the life of young people. I now want to go to bleeding and platelet and thrombotic disorders. And maybe it's good to ask yourself on why has this happened in the UK, this 100,000 Genomes project. And I think the previous chief medical officer, who's called Dame Sally Davis, had a very clear vision, and you might be interested to read her Generation Genome document that is freely available on the web, that very much gave that the message that genomics is not something for tomorrow or the next decade. It is here today. Why? Because it gives better prognostication, better treatment choice, and it allows to explore new treatment options. So can I take you in that space by focusing on one disorder of the smallest cell in the blood, the platelet, the cell that makes the blood clot, but also prevents us from bleeding. It is this cell that if we look in the UK Biobank data, this collection of 500,000 people, it shows that you can measure the volume of the cell very simply by a CBC analysis, and the count of the cell. And you see data of 250,000 males at the left and 250,000 females at the right. And Ernest and I, with our clinical colleagues, agreed that we would enter into the whole genome sequencing program. Patients were presented to hematology clinics across the country with platelet counts below 120 or platelets that had a volume larger than 12.5 femtoliters. And you can say these people this also sit in the UK Biobank collection, but there the consent doesn't allow us to go back to them. So here you see, having worked on these patients with small platelets, with a low number of platelets, or patients with very large platelets, that there is already an immense harvest of new genes that have been added over the past 7 years to the list of candidate genes that need to be surveyed by a MDT. And the publications that underpinning these gene discoveries are shown at the bottom of this slide. So we set up a gene panel test. We did run the gene panel test truly on a global scale and pulled in samples from a large number of people to sequence them as shown at the right on this thrombogenomics gene panel test that included 100 genes that were focused on bleeding, platelet, and thrombotic disorders. So to recap, in the discovery arm, nearly 1,200 patients that got whole genome sequencing. In the diagnostic arm, nearly 2,500 patients that got a gene panel test. At the right, you see that 1 and 2 patients get a conclusive molecular diagnosis. Now, is this important that we assign these diagnoses to patients? Does it inform the treatment? And can I round up on that? Here you see that in a group of 412 patients who have a low platelet count with very large platelets, that in 71 patients there were mutations in transcription factors or the receptors for the growth factor thrombopoietin that are several of those are pre-leukemic disorders. If we look at the 122 patients who had cytoskeletal mutations in genes that are involved in cytoskeletal processes, that several of those patients either have deafness or kidney failure, particularly if we pull out DIAF1 and the MYH9 gene. And I won't for time go on to the granular disorders, but what I want to point out is depending on where the mutation in which gene it occurs, you get different accompanying pathologies that should inform the treatment plan of such patients. So to round up, I want to come back to how the interplay between polygenic trait scores and pathogenic variants. And here I share with you data from a paper that is out on medRxiv from Vukovic et al. That shows that after a decade of genome-wide association studies for complete plot ground traits, that we can almost explain 40% of the population variation in platelet count and mean platelet volume. And I've given you one example to conclude on. In this graph at the right, you see a pathogenic variant in one of the proteins of the von Willebrand factor receptor complex on platelets. The people who are having this variant in heterozygosity, so on a single allele, their platelet count is dotted in the dotted curve. But if they have a high polygenic score for low platelet count, they are displayed in red. If they have a high polygenic score for a high platelet count, they are shown in green. And this graph shows 3 messages. First, if you carry a pathogenic variant in heterozygosity at population level, it's not neutral. if you carry the same pathogenic variant in homozygosity, you're present at the clinic because your platelet count is truly far too low, and they often have bleeding symptoms as a consequence. And what you see in the people heterozygous, that you, for this pathogenic variant in glycoprotein 9, is that depending on your polygenic trait score for platelet count, you either can be moved left in the distribution curve or to the right. So to conclude, based on the results of the pilot study, decision has been reached to make whole genome sequencing standard of care. It's rolled out across all hospitals of the National Health Service. Sharing of data between hospitals is essential if we want to reap the benefits of genomics. And particularly want to reach out to you, to our colleagues in Boston, to say international sharing of data is absolutely paramount for gene discovery. Thank you very much.
Thank you, Willem. Ernest, are you able to share your screen?
I have unshared, yeah.
Great.
Fascinating work. Okay, I'll turn it over to Ernest.
Okay, hi everyone. Thanks, Shira, for the invitation to present. Let's see. Okay, yeah, so I'll carry on from where Willem left off, and I'll give perhaps a slightly more quantitative slant to the talk. I've been in Cambridge for the past few years, but just so you know, I'll be moving to Mount Sinai in September. I'll be closer to all of you lot, COVID-19 permitting. So I will be talking about the following four topics. First of all, I'll describe a statistical genetic association method, which we worked on a few years ago, and it's specifically designed for rare diseases. It's called BEVIMED, and that stands for Bayesian Evaluation of Variant of Involvement in Mendelian Disease. I'll then describe the genetic associations with rare diseases that we have found in the data set that Willem has just described. I'll also describe an analysis of UK Biobank participants who have an extreme value of a particular quantitative trait. It happens to be a red blood cell trait. And I'll finish off by describing the strategy that we followed to try to find causal non-coding variants within regulatory elements, so outside of exons of genes. So I'll start with the method. So this method, BEVIMED, has a few key features that make it quite distinctive. So first of all, it models all the variants, all the rare variants in a locus jointly. So for example, a gene. That's very important because, of course, many different rare variants can have a very similar consequence. So in order to have adequate power, you need to share information between different variants within a locus. We model a bipartition of rare variants into pathogenic and benign subgroups. And that's just because the vast majority of rare variants are benign, even if they are within genes. So we need to take that into account. We model dominant and recessive inheritance. We also have a parameter for penetrance of pathogenic allele configurations. What that means is that even if an individual has an elevated risk of developing a rare disease because of the genetics, it doesn't mean that that probability is necessarily 1. So we have a parameter that models that. We also model the disease risk due to other factors, for example, variants in other loci in the genome. And finally, it's very fast and scalable, so it can be applied to very large data sets, well over 100,000 individuals. So this slide gives an overview of the modeling that we've done. So we basically compared two different models, a baseline model and an alternate model, which we denote with gamma equals 0 and gamma equals 1. Under the baseline model, the probability that an individual I is a case In other words, that yi equals 1 is constant. So the risk of having a rare disease is basically independent of the genetics. Under the alternate model, which we denote gamma equals 1, the probability that an individual I is a case is either tau or pi. It's tau if that individual does not have a pathogenic configuration of alleles, which we encode by saying that this function f evaluates to 0, and it's equal to pi, which is higher than tau, if this individual does have a pathogenic configuration of alleles. So f evaluates to 1. So this function f basically tells you whether an individual I has a pathogenic allele configuration. And it depends on a number of different variables. One is the genotypes for that individual. So these are, of course, known. It also depends on a binary vector Z, which indicates the variant's pathogenicity. And that is not known. That's something that we have to estimate. It depends also on the ploidy at the locus, which will typically be 2, but of course it can be 1 if you're looking at a male on the X chromosome. And finally, it depends on the mode of inheritance, which can be dominant or recessive. So depending on these 4 different variables, that function will return 0 or 1, and that will dictate whether a particular individual has an elevated risk of disease, of case status, or not. Now, it's often the case that only particular kinds of variants in a locus confer disease risk. So for example, it could be that only variants in the untranslated region confer disease risk, or only high impact variants, which cause a loss of function, cause disease. And so what we have done is we allow a comparison of different models in which each model incorporates a different class of variants. So in this schematic here, we have all these different exons. Each vertical line represents a rare variant, and the crosses give you the class or the classes for each variant. And we basically compare these different models that include these different subsets of variants. And if it turns out that only one of these types of variant increases disease risk, then this model comparison will increase power, statistical power. And moreover, it will automatically suggest particular etiological mechanisms. So for example, if you can infer that it is only variants in the UTR which cause disease, then that gives you an insight into what might be going on. For example, these mutations might disrupt binding of a transcription factor. So just to illustrate the types of summary statistics that we get from this method, BeviMed, I'm showing here the top results, where we rank each result by the posterior probability of association. Just going back to the original slide here, that means what is the probability that gamma equals 1? That's the thing that we're most interested in. And, after that, we're interested in Z, which tells you which variants are pathogenic. So here we've ranked by the posterior probability of gamma in a comparison of 112 unrelated cases with thrombocytopenia and thousands of individuals who do not have thrombocytopenia or who are not known to have thrombocytopenia. We have the posterior probability of association next to the gene name. We've then got the modal model. So we have inferred the class of variant responsible, and we've also inferred the mode of inheritance. So we're not just conditioning, we actually infer the mode of inheritance. We have the estimated number of cases explained by these variants, the estimated number of pathogenic variants, and then these are the number of variants on the right-hand side, which were included in the modeling. It turns out that actually all of these genes are known in the platelet rare disease world. So these are all true associations. There was one that caught our interest though, which was the one with GP1BB, where there's a very high posterior probability of association. And we inferred a dominant mode of inheritance, which is different from the typical disease that is associated with GP1BB mutations, which is Bernard-Soulier syndrome, which is a recessive disorder. So using this approach, we were able to actually show that heterozygous variants in GP1BB cause thrombocytopenia as well, which is, it's not as severe as the macrothrombocytopenia in Bernard-Soulier syndrome, but it's nevertheless present. We wrote this up in blood. let's just zoom in on one of these results. Let's look at the top result, which is ANCAR-D26, because this will show you why this method is particularly powerful in certain circumstances. So here we have a probability of association very close to 1. The modal model is 5' UTR and dominant association. So that is where the greatest posterior mass is located. Here we see a diagram illustrating the structure of this gene, ANCAR-D26. The vertical lines represent rare variants. The crosses represent the classes of the rare variants. And underneath, the pink boxes represent the carriers who are cases of these variants in heterozygosity. And in light blue, we have heterozygous controls. And we also have one homozygous control over here and over here. So the thing that we have inferred is the posterior probability of association, of course, also which variants are likely to be responsible, but also the individual per variant pathogenicity. So this is the posterior probability that Z equals 1. In other words, that a variant is pathogenic. And you can see that we've only inferred a high probability of pathogenicity for the variants in the 5' UTR. All the others are very close to 0. And that's despite the fact that there's actually a heterozygous case who carries a high impact variant in this gene in the first exon. But because the vast majority of the alleles that are carried by the cases are in the UTR, we infer that only the variants in the UTR are responsible for disease. And when you look at it that way, you can see that there's a very strong enrichment of the cases in the UTR compared to the rest of the gene. Now consider an alternative approach where you had taken all of these variants together across the entire gene. Then what you would see is basically a lot of blue and not much pink. And therefore the posterior probability of association would have been much, much lower because the enrichment of cases within particular subsets of variants would have been hidden away. Here's another example. This is the result of comparing 3 cases with Roifman syndrome with thousands of individuals who did not have Roifman syndrome. And we inferred a high probability of association with variants in this non-coding gene, RNU4-ATAC. And the modal model here is moderate, recessive, which means that in this case, is any type of variant, but under a recessive mode of inheritance. In this case, we have compound heterozygotes. These are shown with these connecting lines underneath. And what you see in the bar plot above is that the posterior probability of pathogenicity is in general higher than what we saw before. And that's because most of these variants have not been seen in homozygosity, so we can't be sure that they're not pathogenic. But there are a few variants that have a very high probability of pathogenicity, and these are the ones that have been seen in compound heterozygosity in the cases or in homozygosity. There's one homozygous case over here. So you see, you get a very rich output from this method. So what we did then, having developed this method and written it up and published the software, we decided to apply it on a very large scale to the data set that Willem described. So the way that we did that was that we used the HPO data that Willem talked about to define case control groupings. So we had a series of logical rules that took the HPO data and then assigned individuals to case groupings. And here are a few examples of these case groupings: retinal dystrophy, antibody deficiency, thrombocytopenia, and so on and so forth. We had a few dozen of these groupings. And we then tested each gene in the genome for genetic association with each of these different groupings. So for example, for retinal dystrophy, we would go gene by gene and compare all the cases with retinal dystrophy to all the other cases. We would do the same thing for antibody deficiency, thrombocytopenia, and so on and so forth. And we would then follow up results with the highest posterior probabilities. So this figure illustrates the results of that analysis, where we've looked at all of these different case control groupings, gone through all the genes, and we're showing all of the results where we have a posterior probability of association that's greater than 0.75. And they're ordered from top left to bottom right. Each point gives you the posterior probability of association. On the left, we have the gene names, and then on the right, we have the name of the case control grouping. You can see here pulmonary arterial hypertension, pulmonary veno-occlusive disease, intrahepatic cholestasis of pregnancy, retinal dystrophy, etc., etc., etc. So the dots that are closest to 1 are highly confident, and as you go down, they become less and less confident. We have colored these points by whether they were discovered, these associations were known prior to 2015, they were established in the literature. There are 68 of these points, they're shown in blue. And the associations that were discovered since 2015, either by us or by other groups, they're shown in green. And then the unconfirmed associations, of which there are 16, are shown in gray. And overall, we found 95 associations, of which at least 79 are causal. If we just zoom in on one of these case control groupings, which is the thrombocytopenia group, I'd like to show the results for that grouping. So we've got these genes, many of which I showed you earlier. We've got the posterior probability of association. Many of these are known, they're shown in blue, but one of them caught our interest, IKZF5, shown here in red, which had a posterior probability of very close to 0.75. This gene, which is, the long name of it is Icarus zinc finger 5, is expressed quite widely in hematopoiesis. And indeed, some of the members of the family of Icarus zinc fingers are associated with various lymphoid malignancies. But IKZF5 was not known in the platelet field. So this association was surprising to us, and we followed it up. We followed it up by looking at 5 different pedigrees. We had 5 different missense variants overlapping the zinc fingers of this gene. They're shown in blue in the top right panel, and the red points represent the missense variants that were carried by these cases. And the y-axis shows you the normalized conservation score. The lower the value, the more conserved that position. None of these 5 variants had been seen in NOMAD. And if we look at the platelet counts of the carriers, they were roughly half of what you'd expect to see in the general population, which is shown by these histograms. There was one notable exception. One case who had been splenectomized had a correction of his platelet counts, but by and large, they were around 100. We then selected 3 individuals from 3 of the 5 pedigrees for experimental follow-up. We obtained blood samples, we fractionated the blood and into 4 different subtypes of cells, CD4 positive T cells, monocytes, neutrophils, and platelets. And then we compared the expression levels of the expressed genes between these 3 cases and I think about 10 controls. And what we found was that there were practically no differentially expressed genes in T cells, monocytes, or neutrophils, but there were almost 1,200 differentially expressed genes in platelets. And if we looked at the down-regulated genes in platelets, they were enriched for pathways such as platelet activation, signaling and aggregation, platelet degranulation, and so on and so forth. That's shown in panel B here. So this work gives us an initial clue as to the possible mechanisms underlying this new cause of thrombocytopenia. Now, some rare diseases are characterized by reference to a quantitative trait, and we've mentioned thrombocytopenia. There are many other examples: familial hypercholesterolemia, combined variable immune deficiency, and many others. And so we wondered whether, rather than looking at cases, we could look at the general population and take individuals who are extreme for a quantitative trait of this kind and perform genetic association to discover genes which are involved in these kinds of pathologies. So to explore this idea, we defined a red blood cell phenotype in UK Biobank, and we whole genome sequenced 764 participants in the tails of this distribution. So this red blood cell phenotype is a linear combination of the red blood cell count and the mean corpuscular volume. You can see the scattergrams in this slide for males and for postmenopausal females. And the individuals who we selected in the tails are shown in blue for the left tail and in red for the right tail. The underlying grade distribution shows you UK Biobank participants in general. And we then search for genes carrying mutations causal of red blood cell pathology. The first thing we did was to impute a polygenic score of this trait based on published genome-wide association studies of red blood cell traits. And then we looked at the distribution of this polygenic score in the left and the right tails. And what we found was that this score was left shifted in the left tail and right shifted in the right tail, as you can see by these box plots, which indicates that polygenicity certainly plays a role in pushing some of the participants into the tails of this red blood cell trait. However, these shifts were not as strong as predicted by modeling, assuming that the polygenic score is the sole reason for tail status. And indeed a GWAS of an ordinal outcome where we coded the left tail, the unselected group, and the right tail 0, 1, 2 did not reveal any novel associations. So that suggested that rare variants, which are not incorporated into the polygenic score, might be playing a role for a subset of the participants. So we then treated each tail group as a set of cases in a bevy meta-analysis, the same as we did with the rare diseases earlier. And what we found was that a number of genes had a posterior probability of association that was reasonably high. At least 3 of those are almost certainly causal because of the known biology, HPV, TFRC, CUX1, and ALG1 is likely causal. So what this analysis tells us is that we can actually learn about rare diseases by looking at a relevant quantitative trait that can be measured in the general population and looking at the extreme, the genetic determinants of tail status in the extremes of the distribution. So Willem talked about the proportion of the genome that's covered by genes, about 2%. And of course, there's a lot going on outside of the genes as well. But most of the analyses that have been done and that we presented so far involve only the exonic or the intronic regions of genes. Genes have known locations. They are transcribed into RNA. If they're coding, they're translated into proteins. But the regulation of genes is dependent on regulatory elements. And regulatory elements are cell type specific. So the locations are not known perfectly. And their relative importance in regulation is also rather uncertain. So for example, some CRISPR studies have been done in which these elements are deleted one by one. And in some cases, you find that there's absolutely no effect. Nevertheless, there are known examples where even an SNV can have drastic effects on expression and consequently on phenotype. So we searched for mutations in regulatory elements that might be causal of rare diseases. And the first thing we had to do was try to localize these regulatory elements in disease-relevant cell types.
Hi, Ernest. Sorry to interrupt. I want to try to save a few minutes for questions. I don't mean to hurry you up, but do you think we could save a minute or two at the end just to address people's Q&A?
Yeah, sure. Should I just wrap up in 1 minute?
Yeah, thank you.
Okay. So the long and short of this is that we identified regulatory elements using a newly developed method that looks at H3K27 acetylation ChIP-seq and ATAC-seq coverage. We look for a particular pattern of peaks, which is highlighted over here. And we use that to identify the locations of regulatory elements in 5 different cell types, which are relevant to various different hematological diseases, which are shown at the bottom. And having localized these elements, we then searched for mutations in these elements in cases who had a disease that was linked to these cell types. And I was just going to give two examples of this. One is a deletion of an enhancer for GATA1 that also takes out the open reading frame of HDAC6 and results in a composite phenotype comprising macrothrombocytopenia and neurodevelopmental problems. And that's due to decreased expression of GATA1 and an absence of HDAC6. And the other example I was going to give has to do with the thrombopoietin receptor encoding gene, MPL. We found an SNV in a regulatory element that was megakaryocyte specific, and it was in compound heterozygosity with a deletion, and consequently the patient had an absence of MPL on one allele and a 50% reduction in the other allele, leading to 25% overall production of MPL. And this caused a thrombocytopenia, which was not quite as severe as you normally find if you have no MPL, which is called chronic amegakaryocytic thrombocytopenia. So these are just two examples of causal mutations in regulatory elements that we have been able to find. And I'm happy. Yeah, that's all from me. I'm happy to look at the Q&A now.
Thank you so much. So I think I'll read the questions from the audience, and you guys can answer it. I'll start off. Matt Warman asked Willem, are you suggesting that whole genome sequencing supersede panels, or that panels still be a first step followed by whole genome sequencing if panels are negative?
No, I think there's a transition for us. You can't introduce whole genome sequencing in 250 hospitals overnight. But it's very clear that we are determined that over a 24-month period, everything will be moved to whole genome sequencing, except for the relatively frequent rare disorders like hemophilia A, where you might stay gene specific. So answer is panels are out, whole exome sequencing is out, we go to whole genome sequencing.
Thank you. Zoltan Salezi asked, what's available in terms of data for the wider research community?
Ernest, do you want to take that one?
What's available in terms of data to the wider research community? Almost all of the genetic data have been deposited on the European Genome Phenome Archive. There is some degree of access control for obvious reasons, but any researcher can apply for access and obtain genetic, but also some high-level phenotypic data. The epigenetic data have all been released through the EGA as well.
Great. And are all of these broadly consented? So any researcher with any disease focus could leverage those data?
Yes.
Great, thank you. Linda asked, are mutations in these genes somatic or germline variants? I think, Ernest, this is when you had the ANC RD26 slide up.
Yeah, these are all germline variants.
Thank you. Vidhu Thacker asked, has it been possible to validate the variants identified in this method in individual patients?
Yeah, so maybe good to take that. In the first years, every variant that was observed by whole genome sequencing had to be validated in a clinical genomics laboratory. But bottom line is that we have had minimal to almost no discrepancies for single nucleotide variants and for indels, but structural variants obviously need confirmation by an orthogonal technique, but increasingly Oxford nanopore.
Interesting. Mary Carmack asked, can you talk a bit about who research and clinicians have access to the genomes and phenotypic data and other limits to this access? I think you already covered that question.
Yeah, and by the way, the exact details on access are given in a data accessibility statement in the paper. So if you go to the bioRxiv paper, you'll find the information there.
Thank you.
The reason it's a little bit complicated is because it's there are, for example, UK Biobank participants that have different consent than NIHR participants and so on. So there's all the details in the bioRxiv paper.
Of course. Thank you. Let's see. Liang Sun asked, you showed several novel non-coding variants in your presentation. I'm wondering whether we should focus on more non-coding regions for whole genome analysis. Okay.
I think we certainly should. The diagnostic yield still remains quite low overall. Not all of that will be accounted for by non-coding mutations, that's for sure. But there are estimates, for example, from neurodevelopmental disorders showing that I think it was around 4% of the unexplained cases with neurodevelopmental disorders can be explained by SNVs, only SNVs in regulatory elements. So there's definitely work to be done there, yes.
Fascinating. Mary Carmack asked, what's the format for the phenotypic data that was contributed? Is this all in HPO terms?
Yes, it is. Yeah, if you seek access through the processes outlined by Ernest in the bioRxiv paper, you can get deeper access to the data by putting in a slightly more expanded application, and it will be in HPO terms.
Are there additional phenotypic data that are collected, perhaps coming from any clinical records, or? Yeah,
two things is for the 100,000 Genomes project and going forward, twice a year, the data are uploaded with hospital information statistics, and with by the summer we hope with prescription data, and that will continue until someone dies. So that is for the next 60 years or for the next 6 months. Yeah.
Wow. Another person asked, NGS panel out, whole exome sequencing out, whole genome in for rare disease testing. In the future, do you see the same direction for somatic testing?
I think the commitment to do cancer by whole genome sequencing is definitely a move in that direction, but it is going to be done at a lower speed than the rare diseases. The main reason is you would like to have bigger depth for more sensitive calling of somatic mutations, and hence the price of whole genome sequencing has to come down a bit further.
Absolutely. Do you have any plans for transposable elements or large deletion analysis?
We did call and analyze large deletions. We also called a wide array of other types of structural variant. Only the large deletion calls were reliable enough to be used in statistical analyses. However, the other types of variants were used on a case-by-case basis, and there were some, I think, almost 10 complex structural variants which were reported back. We also employed long read octo nanopore sequencing in some cases to resolve individuals who had one coding mutation, one coding variant, but there was a second variant which was likely to be missing and it hadn't been called. For example, in cases where there was evidence of a complete absence of protein expression, we only had one identified allele. And in several cases of that kind, we were able to identify strange alignment patterns in the short read sequencing, which allowed us to do targeted long read sequencing, which then we were able to use to resolve the causal variants. One was an SVA retrotransposon, and another one was a rather complicated, well, I think it was a tandem repeat of some kind. So yes, we have called them. We have also used other technologies to call them more reliably, but it's not, I think, yet that straightforward to include them in statistical analysis, because you would require a very, very high reliability across all the participants to be able to do that.
Makes sense. Dana Woosman asked, who would be eligible for whole genome sequencing covered by the NHS?
So maybe I should take that one, is that before COVID hit, there was a rollout program set up for in 3 waves, and there is a national testing catalog that highlights the rare diseases that were wave 1, what are particularly the neurodevelopmental, the extreme severe diseases where early intervention can help. And in wave hematology, where Ernest and I sit, and immunology is in wave 2, what's expected to go live in December this year. So eventually, once wave 3 is completed, nearly all rare diseases will be in and will be eligible.
Will Renthal asks, do you have a sense for what cell type specific enhancers are more likely to be disease causing than enhancers, whether cell type specific enhancers are more likely to be disease causing than enhancers that are shared across cell types?
So I would imagine shared ones are more likely to lead to developmental disorders, but cell type specific ones might be more likely to be involved in the more narrow diseases. I would guess. I don't know what Willem thinks.
Yeah, I think it is. It's an exciting question, and I think it is. We need reasonable, deep clinical phenotype, as Ernest pointed out, to have reasonable confidence that a disease is a narrow organ-specific disease, or whether a disease is more across multiple organ systems. And so it puts some pressure on the detail of clinical phenotyping. Yeah.
Pankaj Agarwal asks, what percentage of case do you think will be missed by whole exome sequencing that were diagnosed by genome sequencing?
Over to Ernest. He has been running that analysis on behalf of
Yeah, I think it's a very interesting question, which is which is not easy to answer. But we did do quite a comprehensive comparison of whole genome sequencing with 4 different whole exome sequencing data sets. So they included 2 data sets from Colombia, UK Biobank whole exome sequencing, and also interval whole exome sequencing. And we took all the known variants in the human gene mutation database, and we looked at the distribution of coverages. So not just the mean coverage, but how the coverage varies from sample to sample at all of these different positions. And what we found was that there is much, much greater variability in coverage in whole exome sequencing than in whole genome sequencing, which means that even if a large proportion of the time you'll have enough coverage with whole exome sequencing, you're much more likely to find that in a subset of samples, you have insufficient coverage. And we have quite detailed figures on this in the paper. It's an extended data figure 7, and that's on bioRxiv. So I would direct you to that to get a sense of the difference between the 2 approaches.
Thank you so much. I think that is all of the question and the answers that we have that are still open, that haven't been answered. Thank you again. This was really incredible, and we've been getting feedback already from some of the community appreciating this talk and the content. So thank you so much, Willem and Ernest.
Pleasure.
Invitation, pleasure.
Thanks. Okay. Bye.
Bye.
Bye.